Good afternoon, and I'm pleased to be here today to provide this update on ViaCyte's progress developing a stem cell-derived islet replacement for diabetes. ViaCyte is a clinical stage company with the potential to develop a functional cure for type 1 diabetes, thus disrupting the multi-billion-dollar diabetes management industry. We have 3 product candidates in development. 2 of those are in clinical stage, and 1 is approaching the clinic next year. And each of them, it could be viewed as first generation, second generation, third generation products. They all leverage the same core biologic, which is pancreatic cells derived from stem cells. We refer to those cells as PECO1. And importantly, last year, we demonstrated through the collection of clinical data that these PECO1 cells, when effectively engrafted, are able to generate functional beta cells in type 1 diabetes patients. And that's really important because cell therapy as an approach for treating T1D already has excellent proof of concept in the form of islet transplantation data. So developing that with a stem cell source addresses multiple issues with the insufficiency of cadaveric donor pancreases. We have clear paths to regulatory approval with very clear clinical endpoints. And over the years, we've amassed a dominant intellectual property portfolio, currently over 700 issued patents worldwide. And we've been now working with world-class partners in CRISPR Therapeutics and W.L. Gore and Associates, supported by a Tier A group of investors, Bain Capital, RA Capital, TPG, and Sanderling. And importantly, over the next year, we expect a number of critical clinical and preclinical milestones. So type 1 diabetes is a disease that creates enormous burdens for both patients and families, despite much technology that's been applied to this condition for delivering exogenous insulin. So still, patients need to count their carbs with every meal intake, measure their blood sugars, multiple times a day, even with continuous glucose monitoring. And despite all the advances in technology and innovation over the years, there's still just an enormous burden, both physically and thoughtfully, for patients and their families. But hope is on the way. Importantly, cell therapy as a preferred approach to treat type 1 diabetes has advanced over the last 2 decades in the form of cadaveric islet transplantation. And this really has shown a proof of concept of this functional cure, which is shown here with these daily continuous glucose monitoring traces, both before and after transplantation. And what you can see is that it takes the daily fluctuations in glucose and these islets, when enough of them are effectively engrafted, can completely normalize blood sugar without any use of blood sugar monitoring or insulin injection by the patient when it's at its maximal performance. But the limitations here are insufficient number of donor pancreases, as well as variable quality and especially timing of their availability. So that over the last 2 decades, there's really only been about 2,000 patients that have been treated with islet transplantation. And ViaCyte's approach is to use stem cells as the source material, but essentially achieve that same outcome of a functional cure. And importantly, a proof of concept has already been established of our cell therapy in that it can differentiate in vivo in patients to produce functional islet cells. And that's what's shown here on this slide. On the left, we have a histological section of a graft taken out of a patient after about a year of implant. And it's been stained for insulin-producing cells in the brown stain there. And what you can see is that they're abundant, they're clustered, and that's the histological evidence that the cells can do what we need them to do. On the right-hand side of the slide, we have C-peptide evidence, so detecting C-peptide in the blood of patients as a marker for endogenous insulin production. And importantly, the patients enter the trial with inability to make enough insulin to be detected with this C-peptide assay. So this shows that the graft cells, as shown there on the left, are capable of making functional beta cells and producing enough insulin in the patient's blood to be detected systemically. So with that biological proof of concept achieved, that we know the cells can act as we need them to mechanistically, we're currently optimizing the therapy to achieve clinically meaningful efficacy. And in that first instance, in the middle of this slide, an approvable efficacy time point would be something that is not quite the functional cure that we are shooting for, but notably improving the glycemic control of these patients while still having them use some amount of insulin, albeit probably a reduced amount of exogenous insulin. But improving time in range, eliminating or reducing hypoglycemic events, and doing so without any deterioration in HbA1c or glycemic control endpoints. But the optimal efficacy we're shooting for, and if we're able to achieve in patients what we routinely achieve in our preclinical models, would be that true functional cure. So elimination of use of exogenous insulin while maintaining or even dramatically improving glycemic control. And we have a portfolio of products in development to achieve this goal, and they're partnered with best-in-class partners here. So first off, all 3 of these products use the same biologic, these PECO1 cells derived from stem cells. With the PEC Direct product, we're delivering them in an open device. The opening's designed to allow the direct vascularization of the graft cells by the host, but thus requiring immunosuppression like any other organ transplant to protect against graft rejection, and that makes it most suitable for the highest risk subset of T1D patients. In the middle, the PEC and CAP product is a fully encapsulated device. It doesn't allow any cells to enter or leave, but with that comes a requirement for a dense vascular network on the surface of the device membrane. But immune suppression is not required because patient immune cells cannot contact the cells inside the device. And that opens it up to all patients with type 1 diabetes. And we're working on this product in partnership with W.L. Gore and Associates. And then lastly, PEC-QT, which was an immune evasion approach to dealing with the immune system. And this is partnered with CRISPR Therapeutics. And I will touch on that a little more in later slides. But importantly, all three products are leveraging this common PEC-01 cell product, which last year we did show has proof of mechanism in patients with type 1 diabetes. So PEC Direct, the open device to permit direct vascularization, as shown here on this slide. So this is the same graft. Now the insulin staining is the top panel. And on the bottom, we've stained the graft for CD34, which marks the patient capillaries. And you can see just how densely vascularized this well-engrafted region is. So those beta cells have a very dense vascularization network that allows them to sense blood sugar, release insulin that goes throughout the body. So the clinical progress to date has been to show direct vascularization in the clinic consistently. We know the immune suppression is protecting the cells from rejection. We've held a Type C meeting with the FDA to explore the clinical development path here and importantly, get their input on what we would need for seeking regenerative medicine advanced therapy or RMAT designation. And what they said is that clinically meaningful levels of C-peptide that are correlated with glycemic endpoints would be enough as an indication of substantial evidence for efficacy. And we've shown that the biologic substance, when well engrafted, further differentiates, those progenitor cells differentiate into functional islet tissue. And this has been shown by the histology as well as the C-peptide data that occurs in subjects that were entering the trials C-peptide negative. And we're currently in a phase of ongoing optimization to improve that engraftment, both in magnitude and consistency. And we expect over the next 12 to 18 months, we will optimize that to a level which shows true proof of efficacy with C-peptide levels increased and glycemic control due to the graft being very obvious from those increased levels of C-peptide. Switching gears over to PEC-NCAP. So this is now the product with a fully encapsulated device. And we began clinical study with this product a number of years ago. And what we found is that there was a foreign body response that the patients mounted to the device membrane that precluded effective engraftment long-term. So we turned to our partners, now partners at W.L. Gore, to help us devise a better membrane for this encapsulation device. And over the last 3 or so years, we've been very successful with Gore in using a challenge model, a preclinical challenge model, shown here, the nude rat, whereby new membranes developed and optimized by Gore with us, now being used also in a slightly tuned up device as well, produces levels of insulin that if we were to achieve this in humans, it would translate to a real curative, functional cure level of efficacy. And this is data summarized here in the bar graph from 6 different studies in nude rats. But what we've seen to date is this is a consistently robust efficacy with this new membrane in every nude rat study we conduct. It's over 15 to date. Efficacy lasts long-term, so up to 9 months, which has been the longest we've evaluated preclinically to date, but steady levels of 2 nanograms per mil of C-peptide, as well as histological evidence from these rat studies that confirms dense vascularization at the membrane interface with abundant islet tissue, insulin-positive islets within the device lumens. And we're now at a point where we're transitioning back to clinical evaluation. So just in summary here, we showed that the clinical results so far have been safe and well tolerated. The devices, both the original and the new Gore membrane, protect against immune rejection. We've also seen here that when effectively engrafted, those PEC-01 cells will further differentiate to islet tissue, as confirmed by histology evidence in this PEC-NCAP trial. As I mentioned, we characterized a robust foreign body response to the device. It's unrelated, for the most part, to the cells. It's really a response to the device materials themselves. And in using these clinical insights in collaboration with W.L. Gore and Associates, we've developed novel membranes that can overcome this foreign body reaction in the nude rat challenge model. And we're currently testing that new membrane and new devices in the clinic in T1D patients. And initial results there are very exciting. They appear to confirm what we've been finding in our nude rat model. Switching gears one more time, PEC-QT, or the immune evasive approach to dealing with immune rejection, which we've partnered with CRISPR Therapeutics. And the idea here is a one-time gene editing event in the stem cell line to create a new stem cell line or a modified modified stem cell line that has immune evasive properties. And this is a great collaboration because it leverages the strengths of both CRISPR Therapeutics and ViaCyte. ViaCyte contributing our highly characterized stem cell line, CYT49, that's been active in clinical trials in multiple regulatory jurisdictions. Combined with CRISPR's gene editing expertise, we have now created immune evasive lines that could then be used to differentiate to, in theory, any cell in the body, opening up many different therapeutic opportunities. But our first application will be leveraging the previous history of ViaCyte in making pancreatic cells from stem cells and treating diabetes as a proof of concept for the approach generally. In a progress update here, multiple edited lines have been generated. They remain pluripotent and able to differentiate to pancreatic cells and PECO1 cells. CRISPR Therapeutics actually reported results of the first line last year at EASD. We also have preliminary in vitro data that suggests that these edited cells are immune evasive, that they would abrogate detection by patient-derived T cells. And we've shown in these nude rat models that these edited lines have robust function and glucose-stimulated insulin secretion. And currently, we're embarking on the IND-enabling studies using clinical seed banks of gene-edited cells for further evaluation and proceeding towards our first clinical evaluation of the first candidate. Because ViaCyte has been working in this space for quite a long time and also leveraged via consolidation of Beta Logics, which was a J&J internal venture who was also working on the same application from stem cells for a number of years. Between the two groups, we've now amassed a really dominant intellectual property portfolio. We have over 780 issued patents worldwide, 125 of those in the US, and over 250 pending patent applications. And importantly, the term of these patents extends out a good number of years, at least till 2035. Another strength of ViaCyte is our manufacturing capability. So having 6 years of manufacturing GMP cells, devices, and combination products for clinical use, we have really amassed an experience and a track record there. Over 650 cell-filled devices and over 4,000 devices manufactured annually. And we're doing this now in an expanded cleanroom space, over 6,000 square feet of cleanroom space that meets FDA and EU requirements, including some dedicated space for use of gene-edited cells. We have clinically phase-appropriate quality systems in place, and our space and processes right now are adequate for all of our clinical work, as well as initial commercial estimates for the PEC-Direct product, which treats only a smaller subset of T1D patients. Yet we're not stopping there. We've got active process development activities going on to scale up our manufacturing capabilities such that we can treat many, many patients and capitalize on the promise of cell replacement therapy for diabetes treatment coming from this stem cell source. And with that, I will just end, and you can let your mind wander about what the future of diabetes will look like with successful cell therapy on the way. Thank you.